Hey guys, this is about the francisella tularensis. The francisella tularensis cause tularemia, or you can say rabbit fever. This is a lethal condition if you do not treat the patient. What about the bacteria? The bacteria is a gram negative bacteria. Its shape is Coccobacillus. It means its shape is somewhere between coccus and bacillus. It is a category A bioterrorisms. What is the category A bioterrorisms? That is bioterrorisms and used as a bioweapons by countries. There are some other category A bioterrorisms. These are Yersinia pestis, Bacillus anthracis, and in viruses you can say it is Ebola virus. Why they are category A bioterrorism? Because they will cause high virulence even in a low dose. So there are two characteristics: there is a low dose and high virulence. There is a thin peptidoglycan layer over the bacteria, so therefore the bacteria is a gram negative bacteria. This bacteria is a great lover of oxygen, so therefore this bacteria is an obligate aerobic bacteria. They have capsules and type 4 pili. The type 4 pili structure is used for the adhesions of the bacteria to the cells. As this is a gram negative bacteria, so they will not strain with gram straining, normal gram straining. Therefore, they will take the safranin dye, and all these bacteria convert into the safranin color bacteria. Therefore, they dye with safranin. There are two special features about the bacteria. These are the non-motile bacteria, and other things, they do not form spores. As these bacteria are the facultative intracellular bacteria, so they can survive inside the host cell happily as well as outside the host cell happily. These are oxidase and urease negative bacteria, so they do not have enzymes like oxidase and urease inside. In francisella tularensis, there are total number of 4 subspecies, and these subspecies are the francisella tularensis, Francisella tularensis hola arctica, Francisella tularensis novicida, Francisella tularensis mediacitica. The tularensis subspecies, also known as type A subspecies, and the hola arctica, also known as type B. The Francisella tularensis is the most virulent bacteria and cause lethal pulmonary infection. The hola arctica is a rarely fatal bacteria, and novicida, it is a relatively non-virulent bacteria, and it will only cause problems in severe immunocompromised patient. And about mediacitica, we don't know much more about this bacteria. We don't know much more about this bacteria. You have to know about all these bacteria, and these are the subspecies. The culture of Francisella tularensis. This is a fastidious bacteria, means require enriched media, especially cysteine enriched medium. We have three type of medium, that are chocolate agar, BCYE agar, and CHAB agar. Okay, once again, this is a chocolate agar, BCYE agar, and the finally is CHAB agar. BCYE agar stand for buffer charcoal yeast extract agar, and CHAB agar is just a cysteine, glucose, thiamine base agar. In chocolate agar, chocolate agar we have cysteine plus lysed RBC. And BCYE agar we have L-cysteine, activated charcoal, and the finally yeast extract. And in both these mediums, Francisella tularensis form grayish whitish colony, and in CHAB it will form greenish white round smooth colony. Okay, This is a zoonotic condition and spread through infected animals like rabbit, rodents, and deers. And the vector is ticks, mosquito, and deer flies. And finally, there is an accidental host like man. Sometimes the bacteria survive in the water and haystack, so they are also spread through the haystack. So it is much more common in lawn owners. This is about the little pathology about the bacteria. The bacteria enter in our body and loves to bind with macrophages because these organisms are the intracellular organisms. They are the facultative intracellular organisms. They can survive inside and outside the host very well. So bacteria enter inside the macrophages through the process known as phagocytosis. And these bacteria are present inside the membrane bound vesicles known as phagosomes. Normally these phagosomes bind with lysosomes, but here there is no such binding, and the bacteria come out from the phagosomes and start divisions to increase its number in the cytoplasms and increase and finally burst, and all the cell come out from the macrophages and spread through blood and reach to the different organ and destroy them. But the question arises how bacteria survive the harsh environment in our body. And answer is easy. What is that answer? The answer is that they have lots of what is that? Virulence factor. So first is first, let's discuss about the bacteria. These are the encapsulated bacteria. That means they have capsules. And the second thing is the LPS, lipopolysaccharide. Normally, LPS attached to the macrophages TLR4 receptor. So these are TLR4 receptor in the macrophages and activate the macrophages to release chemical mediators like cytokinin, nitrous oxide, and proteases. But there is a twist. This capsule also prevents the binding of LPS to the TLR4 receptor. Therefore, macrophage is not activated. Oh, there is an interesting thing. Macrophage is not actually activated here, but rather they also have type 4 pili. So there is a pili that are known as type 4, and type 4 pili can bind to the macrophages and it is not activated but cause macrophages to phagocytosed. So they do not activate the macrophages. Rather, only thing is that the macrophages phagocytosed the bacteria. And inside the macrophages, now there are a lot of process. So let's discuss what happens inside the macrophages as the bacteria get inside the macrophages through phagocytosis and present in the phagosomes. As the bacteria present inside the phagosomes releases a chemical compound that is known as acid phosphatase, or in simple term you can say it as phosphatase. Okay, so what is the function of the phosphatase? It just inhibits the fusion of the phagosomes to the lysosomes. And what will happen then? If there is no fusion of the phagosomes and lysosomes, the bacteria can survive easily and does not degrade it. And all the bacteria come out from the phagosomes into the cytosol. And what happens inside the cytosol? Binary fusions and increase its number. As you are in the higher classes, you should know another process. What is that? The bacteria also have siderophore. What is the function of the siderophore? The siderophore will snatch the iron from our body. Okay. What is that? Snatch iron from our body and use iron for its growth. It means the bacteria have to be dependent on the host cells. So these are actually help in the growth of the bacteria. So what is actually siderophore? Siderophore is a small molecule. It is a small molecule present in bacteria, and it is a iron chelating agent. It will bind to the iron. There are some bacterias. There are some species of a bacteria. Which bacteria is also francisella turarensis? They have type 4 secretion systems, and this is also helping them to survive inside the macrophages and increase its number through the process known as binary fissions. As the macrophage is filled with and they burst and release inside the circulations. So they get enter into the blood systems and flow to the different part of the body. Then, what is the clinical manifestations of the tularemia caused by the francisella turarensis? On a broad way, you can divide into the localized and systemic. But we have to be very fine about this. We can divide it into 6 forms, and on the basis of the site of inoculation, or in other say that the route of transmissions of these bacteria. So you can divide it into 6 forms. And what are these 6 forms? It's very slowly you should know. These are the ulceroglandular. The next one is the glandular forms. The third one is the oculoglandular form. The fourth one is the oropharyngeal form. And the fifth, sixth is interesting. The fifth one is the mnemonic form, and the sixth one is rare. This is known as the typhoidal form. So the first thing is first, it is the ulceroglandular form and the glandular form simultaneously. And it is the most common route through which you can get infections. And these are the spread through the insect bites, for example, ticks and flies. In the ulceroglandular form, the mostly affecting skin and the lymph node. As you can see in the figure, you're having a swollen lymph node. And in the glandular form, there is no skin involved, only lymph node is involved. So ulceroglandular means skin and lymph node both, and glandular form only lymph node. And in the skin, you will see there are some erythematous rashes. And these erythematous rashes are the tendous pruritic papules. These are the papules, and over a period of a time, these papules are going to converted into the black base ulcers. So they are going to convert it into the black base ulcers. Okay, so the next one is the oculoglandulars. What is oculoglandular? These are spread through your eyes, or in a very easy way, you can say this is spread through the conjunctiva. There may be 2 routes it can spread. The first, when you have a contaminated finger and you rub your eyes with that fingers, and the next, they flow in the air carried by the dust particle. It cause redness in the eye, cause pain in the eye. So the first thing, it is the red eye. Next is pain. The third is periorbital edema. There is a swelling around the eyes. And the fourth one is the photophobia. If you pop up the light, you will feel irritated. Okay, so the next one is the oropharyngeal. So these are spread through the contaminated food and waters, and mainly affecting your throat and mouth. So what is going to affect? They affect throat and mouth. Okay, in that diagram you can see there may be a throat problem like throat pain. In mouth you can get ulcers. You may also get vomitings, and one of the also problem, there is inflamed tonsil, so it causes tonsillitis. And there is also the cervical, especially the cervical lymph node swelling. It will cause painless ulcers in the lymph node also. Okay, so the next one is the pneumonic forms. These are spread through the direct inhalations of the bacteria. Okay, so they directly inhale through the bacteria and cause problems like pneumonia. As we know that pneumonia is of 2 types, primary pneumonia and secondary pneumonia. In primary pneumonia, the first site is your lungs. First time the lungs is affecting. Okay, so secondary pneumonia is that it is a secondary site, means the first site is blood, and through blood it's cause lungs problem, and that is pneumonia. Okay, so there is also a result. There is a very rare person to person transmission, so there are less chances to person transmission. So what are the other symptoms? You may have dry cough, chest pain, and the last one is the difficult breathing, or dyspnea. Okay, so the next one is the typhoidal form. is that any way of entry but cause problem of systemic febrile illness. For example, you may have high fever, may have vomiting, may have diarrhea, or what is the other problem? You may have enlarged liver, so hepatomegaly. Also, the typhoid also cause secondary pneumonia. Okay, so they spread through the blood. If they contaminate through spread the blood and then reach to the lungs. Okay, so one thing common in all the six forms is that the initial things is that the patient present non-specific symptoms. What are those non-specific symptoms? These are the fever along with chills, malaise, arthralgia, anorexia, and the chest soreness. Okay, so the last things is the diagnose. How can you diagnose? So there are 4 ways to diagnose the bacteria or patient, particularly not bacteria. You will not diagnose. Okay, so the first thing is the latex agglutinations, in which you will see increased titer of IgM and IgG against the bacteria after 2 weeks. Next is the culture. As I already told you, the culture will take 2 to 3 days, and there are 3 ways to culture. So what is the sample? The sample may be your lymph node drainage. The sample may be sputum if you have a pneumonic like condition, or maybe blood. So the third is the PCR. It's called the polymerase chain reaction, in which you will find DNA of the bacteria. And the last one is a direct fluorescent antibody test.